Welcome to the Explainer. Inside pretty much every single one of your cells, there's this incredible master blueprint for life, your DNA. But here's the catch. That precious code is locked up tight inside the cell's nucleus, almost like it's in a vault for safekeeping. So how does the cell actually use those instructions to build, well, everything? That's the big question we're diving into today.
So here's our game plan. First, we're going to meet the genetic messenger. Then we'll get to know the anatomy of a gene, which is kind of like the stage where all this action happens. And finally, we'll watch the whole process play out in 3 acts: initiation, elongation, and termination.
All right, so before we jump into the how, we really need to understand the what and the why. Think about it. The cell cannot risk damaging its original DNA blueprint. It's just too valuable. So what it does is it makes a temporary, portable copy of just one specific instruction. And this is where our genetic messenger comes into play.
And this whole process of making a copy, it has a name: transcription. This is the absolutely crucial first step in reading a gene. The cell's machinery finds the exact right spot on the DNA and carefully copies the information from just one of the DNA strands into a brand new molecule called RNA. It's kind of like a scribe carefully copying a single recipe from a giant ancient cookbook. But hold on a second. DNA is a double helix, right? It's got 2 strands. So why only copy one? I mean, wouldn't it be way more efficient to just copy both at the same time and get it over with?
Well, as it turns out, the cell has some really good reasons. First off, the 2 DNA strands have different complementary sequences. So if you copied both, you'd get instructions for 2 totally different proteins, which would just be chaos. And second, the 2 RNA copies would be complementary to each other. They'd immediately stick together, kind of like magnets, and form a double-stranded RNA molecule. The problem is, the cell's protein-building machinery just can't read that. The message would be silenced before it ever got delivered. So here's the bottom line: 1 strand, 1 clear message.
Okay, so the cell knows to copy just one strand. But where on that super long DNA molecule does it start, and where does it stop? To figure that out, we need to take a look at the anatomy of a gene. It's the stage for our whole molecular play.
This whole thing you're looking at here is called a transcription unit. You can think of it as a complete instruction packet. You can see the main players, like the RNA polymerase enzyme, interacting with specific parts of the DNA. So let's break down the 3 key regions that make this whole thing up.
Every transcription unit has 3 essential parts. First, there's the promoter. This is basically the big start here signal. It tells all the machinery where to begin. Then you have the structural gene. This is the actual message, the recipe that needs to be copied. And finally, you have the terminator, which, you guessed it, is the stop here signal. See? Simple and elegant. A start, a message, and a stop.
Now within this unit, those 2 DNA strands have very different jobs. The template strand is the one that's actually read by the machinery to build the RNA copy. It's like the negative of a photograph. The other strand is called the coding strand. It's not actually used to make the copy, but, and this is the cool part, its code is an almost exact match for the final RNA message, just with a T instead of a U. So it's like the positive print you get from the negative. All right, our stage is set. We know all the key locations on the DNA. So it's time for Act 1, initiation. This is where the whole process kicks off. And here are our stars of the show. On one hand, we've got the main enzyme, RNA polymerase. This is the master machine that's going to build the RNA molecule. But, on its own, it's a little lost. It doesn't know where to start. That's where its partner comes in, the sigma factor. The sigma factor is like a guide. It's specifically designed to find that promoter start signal and lead the polymerase to the exact right spot. Together, they make up the complete enzyme, the holoenzyme.
So what exactly is that sigma factor looking for? It's hunting for a specific sequence of DNA letters. In simpler organisms like bacteria, which we call prokaryotes, this start signal is a sequence known as the Pribnow box. And in more complex organisms, like us, eukaryotes, it's a very similar sequence called the Tata box. Think of these boxes as landing strips for the RNA polymerase.
And this image just illustrates that moment of initiation perfectly. You can see the complete RNA polymerase, with its sigma factor buddy, has found the promoter on the DNA. It latches on, and then it starts to unwind the DNA double helix, exposing that template strand so it can be read. The process has officially begun.
With the enzyme locked in place and the DNA unwound, we move into Act 2, elongation. This is where the actual RNA message gets written out, letter by letter.
So now, look what happens. The sigma factor's job is done, so it just pops right off.
The core RNA polymerase engine is now free to move. It starts chugging along the DNA template strand, reading the code, and building a new complementary RNA strand which you can see spooling out of the complex. It's pretty amazing.
Okay, let's break that down. After the sigma factor leaves, the polymerase just starts cruising down the DNA. As it reads each base on the template strand, it grabs the matching RNA building block, a ribonucleotide, and clicks it into the growing chain. So an A on the DNA gets a U in the RNA and a G gets a C. The enzyme then forges a really strong phosphodiester bond between each new block, creating the backbone of the new RNA molecule, literally one link at a time.
Now, the polymerase can't just keep going forever, right? It needs to know when the message is finished. So, welcome to our final act, termination.
Just like there was a start signal, there's a stop signal coded right into the DNA. It's called the terminator sequence. When the RNA polymerase hits this spot, everything just grinds to a halt. As you can see right here, the enzyme lets go of both the DNA template and the brand new RNA message it just spent all that time creating.
Now, in bacteria, sometimes the polymerase needs a little extra help to stop properly. It gets assistance from another protein called the Rho factor. The Rho factor basically helps the RNA polymerase see that terminator signal and make sure transcription ends cleanly. And here's one final fascinating little detail. In eukaryotes, like us, one gene usually holds the instructions for just one protein. We call this monocistronic. But in bacteria, they are way more efficient. A single gene can often contain the instructions for several different proteins, all transcribed in one long RNA message. This is called polycistronic. It's a really key difference in how life's code is organized, and it lets bacteria adapt and produce a bunch of related proteins really quickly from a single command.
So, there you have it. The cell has successfully transcribed a gene. It's created a portable RNA message from the master DNA blueprint. The instruction is ready to go. But a written message is pretty useless until someone actually reads it. So the huge question is, how is that message read to actually build a protein? Well, that's a story for another explainer.